Hey, we're continuing with life science and cell biology in a nutshell. We're learning about peroxisomes. On the left, this figure shows a peroxisome with a paracrystalline core and mesophyll cell, leaf mesophyll cell of a tobacco plant. And it's showing the peroxisome's close association with chloroplasts, which facilitates exchange of materials between the two organelles during photorespiration. Photorespiration is the opposite of photosynthesis. Photosynthesis is where oxygen is produced from CO2, while photorespiration is where carbon dioxide, CO2, carbon dioxide is produced from oxygen. In the second figure, peroxisomes shown in a fat-storing cotyledon cell of a tomato after germination in a seed, and the peroxisomes or glyoxysomes um, in these plant or tomato cells are associated with um, bodies where um, lipid bodies of the seeds where fat is stored, reflecting the central role in fat mobilization and gluconeogenesis, which is the production of glucose or sugars during seed germination. So in plant seeds, you have lipids stored in the seed or fat. So this is converted in the glyoxylate reaction, which we discussed earlier, in the process of turning the lipids in the seeds into carbohydrates or sugars while in aiding of the seed germination and the plant to grow or sprout. So the fatty acid chains in the lipids of the seeds are converted in the glyoxylase cycle to two molecules of acetyl-CoA. The peroxisomes use these acetyl-CoA molecules to make succinic acid, which leaves the peroxisome converts the acetyl or the succinic acid into glucose. And this occurs in only plant cells. So this is how fats are converted to sugars in plants. So continuing on, a short signal sequence directs import of proteins into peroxisomes. Proteins and enzymes used in the peroxisomes. A specific sequence of 3 amino acids located at the C-terminus of many peroxisomal proteins functions as an import signal. Other peroxisomal proteins contain a signal sequence near the N-terminus. If either of these sequences is experimentally attached to a cytosolic protein, the protein is imported into peroxisomes. The import process is still poorly understood, although it is known to involve soluble receptor proteins in the cytosol that recognize the targeting signals as well as docking proteins on the cytosolic surface of the peroxisome. At least 23 distinct proteins called peroxins participate as components in the process, which is driven by ATP hydrolysis. Oligomeric proteins do not have to unfold to be imported into peroxisomes, indicating that the mechanism is distinct from that used by mitochondria and chloroplasts, and at least one soluble import receptor, the peroxin PEX5, accompanies its cargo all the way into peroxisomes, and after cargo release, cycles back out into the cytosol. These aspects of peroxisomal protein import resemble protein transport into the nucleus. The importance of this import process and of peroxisomes is demonstrated by the inherited Zellweger syndrome, a defect in importing proteins into peroxisomes, leading to peroxisomal deficiency. So, with cells that contain quote-unquote empty peroxisomes, there can be abnormalities in the liver, the brain, the kidneys, and death can result even shortly after birth. One form of this is due to a mutation in the gene encoding a peroxisomal integral membrane protein, the peroxin PEX2, involved in protein import. A milder inherited defect is a defective receptor for the N-terminal import signal. So most peroxisomal membrane proteins are made in the cytosol, inserted into the membrane of pre-existing peroxisomes. New peroxisomes are thought to arise from pre-existing ones by organelle growth and fission, which we saw the diagram earlier, and this occurs in mitochondria and plastids as well. And they can also arise from the endoplasmic reticulum membranes too. So this diagram shows the peroxisomes and how they can bud into new peroxisomes containing all the enzymes or proteins by budding or fission of the original peroxisome into two. The proteins or enzymes synthesized by the ribosomes, the free ribosomes come into the peroxisomes. The free ribosomes are not on the endoplasmic reticulum, they're just in the cytosol, and they make proteins that are to stay inside the cell, not being exported to the outside of the cell or to cell membranes, organelle membranes, etc. They're just free roaming proteins created by free ribosomes that stay inside the cell where they're produced. So older peroxisomes rise to newer peroxisomes, and proteins are imported. Also, membrane-bound proteins of the peroxisomes, such as phospholipids, can also come from the endoplasmic reticulum, which makes these membrane proteins as well. And these are called polysomes, and they become a part of the peroxisomes, are transported. So endoplasmic reticulum also has a role in peroxisome maintenance. The proteins are in these different ways transported or imported to the peroxisomes, where they function various ways. Unlike self-replicating mitochondria, however, peroxisomes do not have their own internal DNA molecules. Consequently, the organelles must import the proteins they need to make copies of themselves from the surrounding cytosol. The importation process of peroxisomes is not well understood, but appears to heavily depend on peroxisomal targeting signals composed of specific amino acid sequences. These signals are thought to interact with receptor proteins present in the cytosol, as well as docking proteins in the peroxisomal membrane. Thus, the peroxisome membrane contains import receptor proteins. Peroxisomal proteins, including new copies of the import receptor, synthesized by cytosolic ribosomes, imported into the organelle. Presumably, the lipids required to make new peroxisome membrane are also imported. So lipids made in the ER can be transported through the cytosol to the peroxisome and are also various organelles in the cell. Peroxisomes are thought to form from pre-existing peroxisomes by process of growth and fission. As more and more proteins are imported into the lumen of a peroxisome or inserted into its membrane, the organelle gets larger and eventually reaches a point where fission takes place, resulting in two daughter peroxisomes. So peroxisome-specific proteins catalyzing protein import, shown here, and uptake of proteins from the cytosol. yields daughter peroxisome.